Welcome everyone to Chapter 5. In this chapter, we're going to discuss cell signaling and hormonal responses to exercise. In this reference to this particular video, I'm going to introduce you to the neuroendocrine system and the different types of hormones that we'll see with exercise. Before I get started, let's go over some key definitions. First off is, what is neuroendocrinology? Neuroendocrinology is defined as the discipline that studies hormone production by the neurons and the sensitivity of neurons to hormones and the bidirectional behavior between these hormones, neurons, and the endocrine glands. I know this definition is a mouthful, but hopefully it gives us a little bit of a scope of what we're going to discuss and what endocrinology really is. And so within this field of neuroendocrinology, let's get into some of the nuts and bolts. And first, let's define what the neuroendocrine system is. So the neuroendocrine system contains two components. The first is the endocrine system itself, and this is the system that releases hormones into the blood system to get to its target tissues. And so what we're saying is, with the endocrine system contains a series of different glands that contain these elements called hormones. And the hormones are messengers that go from the endocrine gland, say the adrenals that are on top of the kidneys. They release catecholamines into the bloodstream to reach their target tissue. That's what we're saying with this definition. And so the second component of the neuroendocrine system deals with the nervous system. And the nervous system uses neurotransmitters to relay messages from one nerve to another. And we'll get more into this in the next chapter where we discuss the nervous system itself. But keeping in mind that this component of how the nervous system uses transmitters to communicate from one nerve to another is part of the endocrine system. So when we look a little more specifically into the neuroendocrine system, we need to look into this concept called the endocrine glands. And the endocrine glands are the portions of the endocrine system that directly release hormones into the blood. And finally, with this is, what is a hormone? And so hormones are biochemicals that bind to a specific receptor that are designed to exert a specific effect. Previously, we introduced the hormones catecholamines. And these are, again, norepinephrine and epinephrine. And some of the effects that we discussed that they exerted is increases in heart rate and increases in the breathing rate. And this is exact. And so there are three different types of hormones that you'll need to know. And these types are amino acid derivatives, peptides or proteins, and then finally, steroids. And the differences between these three different types of hormones is their structural makeup and their mechanism of action of exerting their effects onto the target tissues. To better understand the mechanisms of action for hormones, we need to look a little bit further into the concept of the hormone receptor interactions. And so what we mean by this is, for every hormone, they have a particular receptor that they work on to exhibit their mechanism of action. An example of this is insulin. And so when we eat and the food is digested, we have an increase in blood glucose levels. And so with this increase in blood glucose levels, it signals the body to increase the secretion of insulin from the beta cells. And so the insulin is released into the bloodstream, and they work on the insulin receptors found on skeletal muscle. And so once they exert their effect on the receptor on the membrane of the skeletal muscle, there's a series of secondary messengers, and so we're not going to get into that exert their effect on a transporter called the GLUT4 transporter. And what these transporters do is they come to the surface of the membrane to bring in glucose into the cell. And thus, by bringing in the glucose in the cell, it causes a drop in blood glucose levels, help maintaining homeostasis, and also revamping the glucose levels within the skeletal muscle. And this is just one of the many different pathways when we talk about the hormone receptor interaction. And so within these different hormone receptor interactions and pathways, there's a couple of key concepts that we need to introduce to help understand how they work. And the first is this concept known as the magnitude of interaction. And what we mean by this is the concentration of the hormone and the number of receptors on the target cell all have direct effects on how quickly and the intensity of the effect of the hormone. The next item is this concept called downregulation. And what this means is when a hormone concentration is very high, then to kind of auto-regulate itself after the effects of the hormone have occurred, then what we have is a decrease in the number of receptors for that particular hormone. And this third concept is called upregulation. And really, upregulation is the direct opposite of downregulation. So when we have an increase of receptors concentration on a cell in response to a low concentration of a particular hormone. And so what we're doing is we're increasing the sensitivity of the target tissue in response to a low concentration of hormone. And we get this increased sensitivity due to the increased number of receptors for that particular hormone. And so what we're having in this is, with downregulation and upregulation, it's a way that the endocrine system auto-regulates itself. And so understanding the hormone receptor interactions will help us better understand how hormone mechanisms of action work. And so there are three primary types of mechanisms that hormones use to exert their effects on the target tissue. So the first is where the hormone works directly on the nucleus of the target cell or tissues to alter protein synthesis. And a great example for this type of mechanism of action comes with steroids. And so how does this work? So with this mechanism of action, what we find is the hormone, and particularly like steroids, they're lipophilic. And so they have the ability to come directly through the membrane without the need of a receptor. And so once the hormone passes through the membrane, it attaches to its receptor inside the cytoplasm of the cell. And once it binds to its receptor, the action it directly affects on the nucleus. And so it causes changes in the DNA through mRNA transcription and translation, which then directly affects protein synthesis. And so within this mechanism of action, once the hormone binds to its receptor, there's no more receptors along the pathway. So this combination directly exerts its effects on the DNA. And so with this effect on the changes of RNA transcription and translation, the changes in the protein synthesis cause direct changes to cellular actions. And this is how this mechanism of action works. The second type of hormone mechanism of action is the activation of secondary messengers via the G protein. And so when we talk about this particular mechanism of action, there are two different types that we typically see in endocrinology utilizing the G protein. The first is going to be what we call cyclic AMP. And the second is through the utilization of the protein phospholipase C. And so let's discuss on how these two mechanisms work. So with both pathways of the G protein mechanism of action for cyclic AMP and the phospholipase C, they begin the same. In this particular figure, we're talking about the cyclic AMP pathway via the G protein. And so what happens is here in the bloodstream, the hormone attaches itself to the G protein. And G protein, its long name is the G protein coupled receptor. Also, you'll see it written as GPCR or G protein for short. And so what will happen is once the hormone attaches itself to the G protein, it creates a cascade effect of secondary messengers. And so the first complex it activates is called the guanosine diphosphate, or GDP. And then from there, the beta and gamma units come off, and then the alpha unit activates the guanosine triphosphate complex. And then from there, it activates the adenylate cyclase, which then in turn activates cyclic AMP, which activates pKa, and then this causes the protein response that we spoke about for the mechanism of action for steroids. Now, the one portion that I want to point out is for the cyclic AMP G protein mechanism, the activation of the GDP and the GTP are the same for the mechanism of action for phospholipase C. And so comparing the cyclic AMP pathway to the phospholipase C pathway, let's put these side by side. So here on the left, we've got the cyclic AMP pathway. And here on the right, we've got the phospholipase C pathway. And so in both of these mechanisms of action, you have the hormone that attaches to the G protein. And we can see that here while I'm highlighting in purple. And additionally, they both activate the GDP and the GTP secondary messengers. And where these two mechanisms of action diverge is the complex in which the GTP activates. Again, for the cyclic AMP, it activates the adenylate cyclase. However, for phospholipase C, the GTP activates phospholipase C. And then the rest of the secondary messenger cascade is significantly different than compared to that of the cyclic AMP. And so this may be a little confusing, but let's sum it up to what I want you to take away from this. And so with these two pathways, we're talking about the G protein secondary messenger signaling pathway for hormones. And so within this, the hormones for each of these pathways attaches to the G protein, again, which is also termed as the G protein coupling receptor. And in both pathways, they activate the same secondary messengers of GDP and GTP. And where the differences occur is the main protein that the GTP activates. And for the cyclic AMP, it activates the adenylate cyclase protein. And for phospholipase C, it activates the phospholipase C protein. That's what I want you to know and take away from these two mechanisms. And moving on to our last mechanism of action deals directly with insulin. And so insulin itself has its own pathway. And let's dive into this a little bit. Insulin has its own special pathway because no other hormone utilizes the insulin receptors. So insulin is produced and released by the beta cells in the pancreas. And then the beta cells release insulin directly into the bloodstream. And once insulin reaches its target tissue, it binds to the globular protein termed the insulin receptor. And so at the base of the insulin receptor, which I'm going to write as IR, it has a kinase called the tyrosine kinase. And once the insulin activates the insulin receptor, it activates the tyrosine kinase that's on the opposite side of the phospholipid bilayer of the cell membrane. And this is what causes the cascade reaction of secondary messengers within the cell. And so we're not going to go into all the little ins and outs of the secondary messengers that are part of this pathway, which I'm highlighting here in green. But what I want you to know is the series of secondary messengers activates the GLUT4 transporters, right? And we discussed this before. And these are carrier proteins that are located within the cytoplasm of the cell. And once activated by these secondary messengers, the GLUT4 transporters come to the surface of the phospholipid bilayer, and they passively transport glucose out of the blood and into the cell itself. And then from there, the glucose goes on into the energy production pathways. So to sum up what you need to know for this particular mechanism of action for insulin, first is insulin is produced and released by the beta cells. The beta cells directly release insulin into the bloodstream, and they attach to this globular protein known as the insulin receptor, or IR, at the surface of the target tissue. The IR then activates this tyrosine kinase, which then activates a series of secondary messengers. This series of secondary messengers activates GLUT4 transporters, which then come to the surface of the cell membrane to passively bring in glucose out of the bloodstream. And what happens is this glucose is coming into the cell, the overall blood sugar levels drop. This is what I want you to take away with this mechanism of action. So there's some key take-home messages I want you to learn with these hormone mechanisms of action. The first is the hormone receptor interaction triggers cell signaling events within the cell. And we discussed that with all three of these mechanisms. The second is the changing of a concentration of a hormone, whether it's the number of receptors on the surface of the cell or the affinity or the sensitivity of the receptor to the hormone itself influences the magnitude of the hormonal effect. And then finally is the hormones themselves bring about their effects by modifying cell membrane transport, either by activating or suppressing genes that alter protein synthesis and the activation of secondary messengers. Those are some of the key take-home messages I want you to take away from this section of the lecture. To sum up what we covered in this video is first, we defined what neuroendocrinology is. Within that, we talked about the basics of the neuroendocrine system. We defined hormones and endocrine glands. Secondly, we discussed the different classes of hormones. And we said this entailed steroids, amino acid derivatives, and finally, peptide hormones. And finally, we discussed the different types of mechanisms of action for hormones. And we said that there are 3 types. The first is the direct effects. We saw this primarily with steroids. The second is through the G protein. We said there are 2 types here, the cyclic AMP and phospholipase C. And lastly, we talked about the insulin pathway. So this concludes our first portion on hormones. I hope you've enjoyed this video, and I'll see you in the subsequent videos. Take care.